My name is Lucas, and I study Biomolecular Science at the Vrije Universiteit Amsterdam. One important aspect of human evolution has been the transition from hunting and gathering to the cultivation of livestock and crops for our food supplies. This has allowed us to build permanent settlements and subsequently led to extreme population growth. As to be expected, larger populations need larger food supplies, hence the agricultural technological development has become a very important aspect of modern society. One of these agricultural developments is soilless agriculture. As the name suggests, it uses another medium to carry nutrients to the crops. It uses less space and water compared to conventional agriculture and can be performed in a controlled and potentially sterile environment. This optimized use of space and water and the ability to have multiple harvests a year have greatly increased agricultural output. In contrast, organic agriculture is deemed a more sustainable form of agriculture that only allows natural resources to be used in terms of fertilization or pest control. Previous research has found that organic soils are more microbiologically diverse than conventional soils. In addition, other research has shown that the human microbiome has been linked to many different diseases ranging from neurodegenerative disease to tooth decay as well as diabetes. The dietary intake of microorganisms may therefore influence the human gut microbiome composition, although currently this is mostly studied in the field of probiotics. Another study has shown that fresh produce intake has been linked to better mental health compared to intake of their canned varieties. Authors have attributed this benefits to mental health to the higher nutritional value of fresh produce compared to their canned counterparts, and have not studied the effects of the human gut microbiome that might have been as a consequence of the microbes alive on the fresh produce. And despite the difference between the microbiological composition of conventional and organic soils, not much is known how this translates to the microbiomes of the conventional or organic crops themselves. So we hypothesize that the growth medium greatly directly influences the crop microbiome and subsequently potentially human health through dietary intake. As organic soils are microbiologically richer, this richness would directly translate to the richness on the crop as well. So to this purpose, we have taken 4 crops: the cherry tomato, lettuce, snack-sized cucumbers, and bell peppers. From each crop, we'll sample the crop itself and the rhizosphere surrounding the main plants. From both the crop and the medium samples, we'll extract DNA for species richness analysis and perform plate counts to estimate the bacterial load on the crops and media by counting the total colony-forming units. As this is an ongoing study, I don't have that many results to share with you yet, although I can tell you that soilless media indeed show lower colony-forming units compared to their full soil counterparts, and that tomatoes are seemingly nearly devoid of any bacterial life. The only exception being organically grown tomatoes that were full of one type of bacterium, but it is likely the same bacterium that farmers themselves applied as a form of organic pest control. In the rest of the study, we'll await the rest of the data on colony-forming units, and as we speak, the DNA extracts are on their way for analysis at an external lab. In the future, we hope to use a model of the gastrointestinal tract to verify that microbiologically rich crops that are ingested can settle and interact with the human gut microbiome. Thank you for watching.